Hi, welcome, welcome, welcome. Hi, Tony. Hi. Wow, you guys are like straight in there. Hi, guys. It's a funny one. You guys have got your exam on Wednesday, and I feel nervous about tonight. I don't want to make any mistakes for you guys, so it's a funny one. Um, Okay, so I've got a whole load of questions, a whole load of content I'm going to go through tonight. So I will go for as long as I can go to try and get as much out. I will go live again tomorrow night as well. So any content which I haven't covered, I will um I will go on again. I know I was sitting here basically going and watching the clock, watching the clock, watching the clock, thinking, should I just go live? I was like, well, it takes me time to set up and nobody will be ready for it. But hi, everyone who's already joining. Welcome. It's so scary that it's so soon that it'll all be over, but it's good at the same time. So, okay. Hi, welcome, guys. So what I'm going to do tonight, okay, for paper 3 is I put out a educated guesses. So I've looked at paper 1. I still have not seen paper 2, okay? So I've been watching and waiting for it, but it's not gone up on the system yet. So I still haven't seen paper 2. So all I've got to go on that is the clients that I've seen already and also the emails that I've got from people. So what I would say is from what I've been told and from what I've seen in paper 1, I've made some educated guesses as to where they might go. So what I'm going to do is I'm going to go through topic areas that have not come up, but do it in a way that shows you the holistic side of things in the connections, because this is what this paper is really about. Do you know how those content that you've learned, all the content that you've learned can be put into like a random novel situation. But what you've got to do is bring your knowledge to the forefront. So I'm going to show you some questions that are not, just to be clear, not OCR questions, the ones I'm going to show you, because you've done them already. You've exposed yourself to them. They're not going to help you now. What I'm going to do is I'm going to help show you where they could go with different styles of questions, but on the topic areas that I believe could come up. I've also got a list that I made and a list that other people have made. So I will start to add in extra bits. But what I might do, so this is live. I'm on here right now and I'll keep going. What I will do is I will do a bit of content, then I'll come back and look at some questions. So the first thing I would ask you guys to do, for those of you who are on here, can you please not just bombard questions? Oh, how did paper 1 go? How did this go? How did that go? Because that's not going to help you, okay? What I want you to do is I want real questions as I'm going on this, but what I would say is if you can, I mean, this isn't like how I'd run my normal actual sessions. Obviously, that would be very restricted, but I want to help you as much as I can. So I want to answer the questions as I go. But it may be that if there's too many questions coming through, that I can't answer everything. So I will come back every now and again and check some of the questions. How does paper 3 work? So paper 3 is obviously going to test you on anything. So they'll always give you graphs, they'll give you information, they'll expect you to apply your knowledge. So it's all based on anything they haven't covered already, as well as what they are going to do is they're also going to not just do the stuff they haven't tested you on, but also do the content that links to topic areas. Okay, so what I'm going to do is I'm just going to get started because there's quite a lot of you in here now. Wow, 59. Crazy. This is like exciting. So what I'm going to do is I'm going to start and I'll come back and start answering some questions. So for those of you who are wanting to ask questions, it's not practical heavy. No, Sam, it's not practical heavy at all. There will be practical parts to it, but they've already put in practicals in paper 1 and paper 2 that I've heard of. So what I would say is let's get going and I'll come back and answer some questions as we go. So I'll keep an eye on the chat. However, I do want to just start covering some content with you. Now, one of the first thing I want to cover is about fatty acids. I don't know why, but I do feel like they're going to bring in something to do with fatty acids because they did a lot on polysaccharides last year and they've done a lot on proteins, but they've not done that much on lipids. And lipids can come in quite a few different areas. So one of the things they could do is they could say that here we have a diagram, and that diagram is showing you your fatty acid, what type of fatty acid is actually shown. So straight away, you should be able to identify the fact that there are double bonds here. And the fact that there's double bonds there, therefore means that we are automatically looking at a unsaturated fatty acid. So you should know whether it's saturated, whether it's unsaturated. In this case, because I've got two of my double bonds there, I can say that this is a polyunsaturated fatty acid. So remember, you will have your carboxylic acid group at one side, and then the carbons are filled and either saturated with hydrogens, or in this case, unsaturated. So this is one way in which they could ask a question. The other thing they could do as well is they could actually give you a bit of information. So they could tell you that they've got some cocoa butter. They're telling you it's filled with triglycerides. They've given you little bits of information. And they are saying that they are made from different fatty acids. So they've shown you 3 different fatty acids that are found in the cocoa butter. So you can see here, you've got linoleic acid, you've got palmitic acid, and you've got oleic acid. And you see something like that and go, oh, I didn't get given any of this information. I don't know. So therefore, what I would say is when you see something like this, note that they can either give it as a diagram like I've done above with the carbons, or they can do it as a diagram as I've shown below, where it's just zagging lines. Now, what that therefore means is every point where there's a bit at the top or at the bottom, that is representing where there is a carbon, okay? So therefore, you can use that to guide you. And what we're seeing here is we're seeing double bonds because I've got 2 lines there. I have no double bonds in the middle, and then I have a double bond here. So I have differences as well as similarities. So therefore, what we can see is we have these 3 different fatty acids, some that are unsaturated, some that are saturated, some that are polyunsaturated, and some that are monounsaturated. So one of the things I would say to you is, as you're going through these questions, do make sure that you know how to get some of the similarities and differences, and I'll leave that for you to see. But it's important that you know carbons, hydrogen, oxygens. They've already given you a question on the multiple choice paper with that. They're both fatty acids, so they both have carboxyl functional groups. So those types of bits of information are just the starting block, because remember, these papers are assessing you all the way from getting that E grade right the way up to the A star. Start at the beginning. So at the moment, you don't need to be talking about the kinks. However, it does state down here about that creates kinks. Now, I do think the kinks are going to come in, but I think the kinks with regards to the unsaturated fatty acids is going to link to membrane structure, because if I'm wrong, correct me if I'm wrong here, guys, but they haven't really assessed you on biological membranes yet. So therefore, they could link fatty acids with the membranes. So one of the things to remember is you have got your glycerol molecule, you've got your fatty acids, you've got your hydroxyl groups, you've got your carboxyl group, and there's an uneven sharing of electrons, which results in a hydrophilic part. Now, that's important to note, because if I've got a hydroxyl group, I know my oxygen and my hydrogen are going to have an uneven sharing of those electrons. Oxygen's bigger, pulls the electrons closer to itself. If I was talking about water, I would therefore be mentioning about the fact that my water molecule has this delta positive, delta negative. So I know if there's an uneven sharing of electrons, And basically, our water molecule is the hydroxyl group. So therefore, we've got two. It's a dipole. We have two polar ends to it. So if we have hydroxyl groups with a glycerol molecule or with a fatty acid, we're therefore going to get that one little part, which is going to be hydrophilic. Now, this is important because when you then look at the hydrocarbon chain, remember what I said before, all those dots are representing where the carbons are. When you look at that part, that is what makes it insoluble. Because those carbons and hydrogens share those electrons evenly, they lack any charge region. And that's going to make lipids insoluble. The more carbons there are, so more than 12 carbons, the more insoluble the molecule actually is. And that will be important if they do ask a question about the function of lipids. So when we look at, don't worry, I am going to bring this up. When we look at whether it's saturated, unsaturated, mono or polyunsaturated, it's important to note where you see those double bonds, but also the fact that you can end up with, as I'm showing in this diagram, these kinks. So here, here, and here are my double bonds, and you can see that it's not giving me a straight line like my saturated fatty acids. Instead, what's going to happen is I'm going to end up with kinks. And those kinks are going to stop things from packing together very closely. And that therefore is going to cause a problem. So I'm going to talk about the membrane structure. What I want to do is I will take you on a journey here, guys. So it's not just about you memorizing that content. If you want to look at that content, go look at my videos on, excuse me, the lipids. Go look at my video on the membrane structure. What I want to do here is I want to help you see the bigger picture so you can see where they're going to do those interlinking parts, okay? Excuse me. So double bonds are going to cause these chains to bend. They're therefore not going to pack together. This is OCR A for those of you who are just joining. And therefore, because they can't pack together quite so closely, they're normally lipid at room temperature. So we tend to say they're oils. Well, if we have a membrane which has got unsaturated fatty acids in it, that is going to result in us having less packing together of our phospholipids. So that when we look at our phospholipids, we know that we've got our glycerol molecule attaching to our 3 fatty acids. A macromolecule is going to give us a triglyceride. So if we can create a triglyceride by creating these ester bonds, removing water, condensation reaction, then what we can do is we can take that glycerol molecule. So here we have my glycerol molecule. And instead of me having 3 fatty acid chains, now what I have is I actually have a phosphate ion that has been attached to it. And now what I have is I've now got a hydrophilic. Please, 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 in your exam, do not just say it's a hydrophilic head. It's a hydrophilic phosphate head. It's got one of those fatty acids that's been replaced by a phosphate group. And so you know those phosphate ions are negatively charged and therefore they are dissolved in the cytoplasm. So that phosphate ion can be added to the glycerol molecule. Instead of you getting a triglyceride, we're getting a phospholipid if we need to make our membrane structures. So then you've got your hydrophobic fatty acid tails. And when you've got those hydrophobic fatty acid tails, what's going to happen is they will form a layer on the surface of water. Now, you know that the phospholipid bilayer is a bilayer because it is creating this barrier which has water on either side. So it's created a barrier in between because what we have is we have a different structure. So let's just look at a question that they could ask you on this. So explain what phospholipids containing more unsaturated fatty acids would do to the fluidity of the membrane. So do you understand that the phospholipid, as we said before, is made up of a phosphate group, a glycerol molecule, and two fatty acid chains? Well, we always draw our phospholipids really nicely like this. We always draw them to show, and I'm doing this really quickly, so excuse my really dodgy diagrams. We do it really nicely as if they're quite straight and they're saturated, but actually they might be unsaturated. And because they're unsaturated, that's going to cause more gaps between the actual membrane. So therefore, if you've got more unsaturated fatty acids, you're going to expect there to be more kinks. So less closely packed. And therefore, that's going to have an effect on that fluidity. So one of the things that you need to remember is, well, what causes the kinks in the first place? Is it enough for me to say kinks? Well, absolutely not. You need to be mentioning in your answer what causes those kinks. It's unsaturated, so it's a double bonds between the carbons. Now, I know I can talk for Great Britain, absolutely, but when I'm writing my answers, I am trying to make them as specific as I can. One, because I hate writing that much, but also I want to make them specific so that I'm hitting the keywords and the marking points that are going to be what the examiner's looking for. So double bonds between the carbons, therefore less closely packed together. So if we were to look at then the phospholipid bilayer, here's one I prepared earlier. Here you can see my phospholipid bilayer that I've got my hydrophilic heads on the outside and I've got my hydrophobic tails on the inside. Now I've colored them different colors to help represent the fact that you have extracellular fluid on the outside, you have your cytoplasm on the inside, and therefore what you have in the middle in this purple region is the hydrophobic region. Yes, to answer the question, thank you, Arthur, for saying that. I have prioritized the topic areas that I believe are going to come up. I do have other stuff that I will cover, but I'm prioritizing. Basically, I'm giving you a heads up of what I'm doing with my students that I believe is going to come up. Now, I might be wrong on that front, but I'm hoping that I'll be able to cover as much of the content for you that will hopefully help you. So if you are looking for specific subject content, you want to do a topic and I'm not covering it right now, and you get bored because I'm just chatting away, go and watch my other videos. They are linked to the specification points and they're in playlist for you for that reason. But right now, what I'm going to do is I'm going to go through what I believe they may ask and I will bring it back through. I don't believe gene manipulation's coming through the whole of it. I don't believe that. I have got a bit that I want to cover because I believe genetic engineering is coming in. Matthew, what have you said? So would the membrane be more fluid or less fluid when unsaturated? If it's unsaturated and you've got more kinks means it's less closely packed together. There's going to be more fluidity of them moving around. So that's going to have an effect on it. But you've got your phospholipids, you've got your proteins also embedded here. So carrier proteins, channel proteins, glycoproteins. What's really important is that it's a fluid mosaic model because the phospholipids and the proteins can move around. Now, this is important because if they started to look at, I don't know, ethanol, and they were looking at fermenters, I was talking about fermenters in one of my lives previously. If they were to talk about fermenters and they were to talk about the brewing of your yeast and the fact that the yeast are anaerobically respiring, they're anaerobically respiring, that's going to therefore start to produce ethanol because we've got the ethanol pathway. If we get the ethanol and too much ethanol is being produced, that is going to disrupt the membrane. And I will link back to this as well, because you should know if we were doing genetic manipulation, so gene manipulation, we need to extract DNA. So if we need to extract DNA, what are we going to do to separate out? Let's think about that. We're going to use ethanol, because ethanol disrupts the membrane, and also it is going to cause the DNA to precipitate out from the water. So it's important you note there's links in lots of different ways. It really just depends what that examiner, the person who wrote the exam questions, has decided for this year's exam. I could go and talk about all the different topic areas based on you asking me one question, because that's what this is about. Biology is that bigger picture. So you should know all of this content. What you're trying to do now is prove that you can apply it to the topic area that they've decided to ask about. So phospholipids and proteins are free to move around. We know it's a mosaic model because there are different proteins, sizes, shapes. I'm going to also talk about that with hormonal communication in a bit, are embedded in the bilayer. So the fact that we have the proteins and the phospholipids able to move, well, we know anything to do with temperature, straight away in my head, I'm going temperature, kinetic energy. If there's low temperatures, less kinetic energy. If there's more high temperatures, more kinetic energy. So if these are moving around because they're fluid, they've got kinetic energy. Even if something is a solid, as it sits, it will vibrate. This is bringing GCSE knowledge into it. So as it's actually sitting, it's vibrating. If I give it more kinetic energy, it's going to move more. If I give it more kinetic energy, it's going to move more. If I give it enough kinetic energy, I can change the state of whatever it is that I'm talking about. So that's GCSE knowledge needing to come forward into this content. So these phospholipids, these proteins, are going to be able to move around. So because they can move around, it means that they are able to function the way that they need to. So we need a way in which we can stabilize the membrane, because our temperature changes. Our temperatures get a bit hotter. I'm getting excited. I'm moving around more. My muscles are contracting. I have ATP. I've got lots of metabolic reactions going on. Therefore, I am going to be generating heat. Therefore, my membranes, those phospholipids, are going to be moving around loads. Well, my cells are not going to function if they move around too much. So therefore, what we want to do is we want to stabilize that membrane. No, I actually mean move around. So if you think of my hands, right? So if this is my hands are being the phosphate heads, and these are the fatty acid tails. What I'm saying is they can do this. So if they're doing this, if they're moving around, okay, they're moving around, they have the capacity to move around, they can just vibrate. If they've got more kinetic energy, they're going to be moving. And as the proteins are moving around, they're actually creating this different mosaic. So if I start to increase the temperature, woo, you're seeing me more than you were before. So if I'm like in there, well, look at the gap. I've now got a big hole. My membrane is more porous. That's a problem. The whole point is this phospholipid bilayer is a barrier. It is creating a partially permeable, selectively permeable membrane. So we want it to be this barrier. And because of that, they need to ensure that there's some form of stability within the membrane. And in this case, yes, this is why I'm doing it on my YouTube, because it automatically saves it so you have access to it straight afterwards, okay? And yeah, so basically, if we're looking at that, we now need to talk about cholesterol. Ooh, lipids. This is part of the lipid content. So this is biological membranes, but it's also biological molecules. So this is two ways in which they can assess you on that content. Do you actually know about cholesterol? Do you know that cholesterol is actually embedded into the membrane? And then, do you know that that cholesterol is going to stabilize the membrane by decreasing its fluidity? So Jamie, you've asked about lipoproteins. So lipoproteins, lipo means lipids, proteins is proteins. So when you look at a lipoprotein, lipids we know are insoluble. Therefore, they cannot be transported in the bloodstream unless they are attached to proteins. And that's what a lipoprotein is. A lipoprotein is where you've got lots of phospholipids or triglycerides with your proteins. So you basically have a very special ball, for want of a better way of describing it, a very special ball that holds the fats inside because they cannot actually be transported in the bloodstream without having that. Now, the lipoprotein part of your topic area, if you're AQA, yes, that's part of it, but OCR, they will give you everything you need to know about the structure. If they go down HDL or LDL, they'll give you information on that one. It's not actually something you have to know for OCR. Yes, you should know the structure of cholesterol, absolutely. So the peripheral proteins are on the outside. An intrinsic protein is embedded inside. So this is important, the difference between the two. Excuse me, guys, I'm still suffering with this cold. Okay, so cholesterol stabilizes the membrane, decreasing the fluidity at high temperatures because we don't want them to separate too much because it's going to let things in and let things out. That's a problem. But if we go too cold, we don't want them to be stuck together. So therefore, the cholesterol actually stops them from becoming too close together because if they become too close together, things can't get across. Things can't move through if those phospholipids are not able to move. So therefore, it's really important to note the different functions of lipids because this is where I think they're going to go. So they may ask about it to do with a energy source. So the fact that the lipid is an energy source, more energy per molecule. If you've got lots of carbons with hydrogens, you are going to end up with a higher energy source because they can be broken down into acetyl groups. Those acetyl groups, can enter at the link reaction, at the end of the link reaction, straight through into the Krebs cycle. So there's more energy that's going to be because there's lots more hydrogens. If there's more hydrogens, those acceptors, your coenzymes going to accept them, take them to the ETC, and therefore you're going to have more ATP being produced. So your energy source for respiration, more energy per molecule. For 100 grams, you get more energy in a lipid than you do a carbohydrate. And that's because there's way more hydrogens there. This is the other part where I'm talking about the membrane formation and stability. So that cholesterol, there's a hydroxyl group on the cholesterol molecule, which is why it sits just up where the heads are. And then you have the rest of it, which is hydrophobic. So the rest of it sits in with the hydrophobic region because it is mainly hydrophobic. So that's one way they could go, looking at that stability of the membrane, looking at why cholesterol is important. Or they might link it because you haven't covered hormonal communications in much detail yet. So therefore, if it is used to actually manufacture your vitamin D, steroid hormones, bile, liver, excretion, your steroid hormones is hormonal system, obviously, and then the vitamin D. So if they're going to do that, this will be where they're going to now take you on a journey and talk about the hormonal system. So it could be a one marker on cholesterol, and then the rest of it is on hormones and how hormones work. But it is important that you know cholesterol is needed to manufacture those hormones. Thermal insulation, reducing the heat loss. Electrical insulation, I'm going to come back to this when I circle back and do neuronal communication. The myelin sheath. Myelin sheath is basically lots and lots of phospholipids that have been laid down again and again and again. So therefore, you are insulating the electrical activity of the neuron. Buoyancy, in the fact of aquatic organisms, keeping them buoyant, and then protection of our vital organs. All of our organs are surrounded by these layers of lipids, and that's so it ensures that as we're moving around, they're not stuck in position. Our organs are moving around with us, and therefore they need to be cushioned against impact. How is it involved? You just need to know is that one of the molecules that is needed with the smooth endoplasmic reticulum, and it will then be joined together to form whatever the hormone may be. So if it's aldosterone, if it were testosterone, if it were estrogen, for example, that's the sort of thing I'd be looking for you to understand. But it won't want you to know any more detail apart from that it's used in the manufacturing of it, and that occurs at the endoplasmic reticulum. What they want to assess is, do you actually know its role and the fact that it is a lipid? The ETC bit that I was describing earlier was basically just talking about the carbons. So if you think about your long chain fatty acids, if we break those long chain fatty acids down, let's say there is, I don't know, 52 carbons in one of the fatty acid chains. If we were to break that down and we therefore end up with our acetyl groups, how many acetyl groups roughly could we get out of 52? We'd have 2 carbons, 2 carbons, 2 carbons. Let's say we get about 20. I know that's not the right number because it's a whole reaction thing, but imagine we get 20 acetyl groups. Those 20 acetyl groups are going to be able to enter the Krebs cycle. So because they've got so many hydrogens, and we know that hydrogens are needing to be taken from a molecule and transferred to the ETC for those electrons to be moving, because the hydrogen atom gets split up into a hydrogen ion and gets split up into an electron, and the electron makes its way through. So it's important you note that linking together, I don't think they're going to go down the route of the ETC, by the way, but just so you are aware, that linking together is where they could go, but I feel like they've done too much on that already. Yes, I did say anaerobic respiration. I do still feel anaerobic respiration, culturing yeast for fermenting. For some reason, something's sticking in my head about that one. But again, this is guesses, guys. I might be right. And if I'm right, amazing. I'm really pleased for you. But if I get this wrong and you only study what I've said, then you may actually be at a disadvantage. So make sure you have a overview of the majority of it. Do lipids form the wax? Yes, yes, they do. Yes, they do. So waterproofing, as I've said here, birds' feathers, puffins, and plant leaves and waxy cuticles. So you definitely need to be aware there's lots of different ways in which they can go with this. This is why I have a feeling they haven't done lipids in a while. So the last kind of big question I saw on this was maybe 4 years ago. So I do feel like it could come back on that. Only cover the stuff you really hate would be my advice to you. Right, where have I got to? Oh, the other thing to note is ethanol emulsion test. I do feel like an emulsion test could be asked because, again, guys, correct me if I'm wrong, I don't feel like you've had any type of quantitative or qualitative testing. I haven't seen the paper. I did definitely feel like cellular control was coming up. I was surprised I didn't go down the lacoperon more or transcription factors, but again, I haven't seen the paper, so I couldn't say. And I don't know the mark scheme. I need to look at the actual question about the Hox genes. The fact that the Hox genes are transcription factors, I do think potentially that one was correct. I did say it would be about evolution and natural selection, but again, I haven't seen the test. Barely in a graph, that makes me think there was a graph. There's lots of graphs for you to analyze in paper 1, which makes me think you've definitely got a graph to draw in paper 3 because you haven't had one yet. No graph representation questions or bio drawings. Yeah, bio drawings, I do feel like it's coming in. They always throw in histology, but I don't know what they're going to do. I think the histology potentially could be your muscles. Yeah, so the cellular control side of things, but I can't tell you yeah, Hox genes are transcription factors. So yes, I corrected myself, Tony, when I was like, oh no, they haven't, but they have, because the Hox gene side of things. I feel like potentially a histogram. I want to show you now. So we're going to go on a bit of a detour, and I now want to show you the types of questions which are assessing your knowledge and application skills when it comes to content like neuronal communication and muscles, because I really do have a strong feeling that they're coming through. How they come through, I'm going to show you lots of different ways that they could ask this type of question. Yes, I definitely think this experimental design is coming up. I said it would either be on photosynthesis or respiration, but because you've got so much on the photosynthesis side of things, it makes me think the respiration and the culturing of yeast could potentially come up. So yeah, so that's the way I would look at it. Right, let's have a look at this question then, and I want to show you where they can give you hints in the question. So they've done it before where they gave a question to do with the statholith, for those of you who've seen all the past paper questions, where they give you a bit of information and then they expect you to then apply your knowledge. So in this case, they're talking about this green lacewings. They're giving you some information. They're giving you its scientific name. They're telling you that it has a sense organ, which is the tympanal organ. It's going to detect sound. It tells you that they've evolved to detect high frequencies. So I got rid of the evolved question because the fact that they've already done an evolved question means we're not interested in that. I don't feel like that will come back up. However, what is really interesting is they then go on to say, when high frequency sound is detected, the receptor cells stimulate the transmission of an impulse in a sensory neuron. So here they've already given us some information. Now, what they're asking us to understand or to prove to them that we understand is the sequence of events that result in an action potential in a sensory neuron. So this is basically them saying, do you actually know your neuronal communication? Do you know how this is going to actually cause an action potential? So the first thing I would say to you all is, what is an action potential? So if you were in my class, I would ask the question, what is an action potential? And some students know it straight away, and they will start talking about minus 70 millivolts, and they will start talking about the fact that the minus 70 millivolts is then going to have a stimulus. Well, at the moment, I'm not talking about an action potential. So right now, all I've spoke about is my resting potential, and then I've talked about a stimulus. And that stimulus is actually going to be depolarizing the membrane. So whatever that stimulus is, that is going to depolarize the membrane. And then if I manage to reach, that stimulus is going to reach my threshold value. That is going to trigger an action potential. So when we're talking about the receptor cell, we need to be saying that the stimulus reaches the threshold value, and this triggers the generator potential. Now, the generator potential is going to be the potential within the receptor cell that is then going to be converted into an action potential in a sensory neuron. Now, we haven't actually mentioned anything yet. We have not spoke about the action potential yet. Tanya, I don't know. It depends how long I can go for. My plan is to at least probably get to about 9 o'clock, and then I'm going to probably crash out. But no, those of you who've been with me for a while will know I'll just keep talking and talking and try and help you. So we'll see how far I get. So my resting potential, minus 70 millivolts. What causes that? What's going to cause that is the fact that we have our sodium-potassium pump, sodium-potassium pump, pumping sodium in one direction, potassium in another direction. We're therefore depolarizing the membrane, coming away from that resting potential. We reach the threshold value, then we are going to know that our action potential is going to be triggered. Now, an action potential is 3 parts. Number 1, number 2, and number, what color have I not used? 3. An action potential is depolarization, repolarization, hyperpolarization, all the way back. So it's important that you note, TS, I'm going to come to this in a second, hold on, because that is the starting point, which will freak out most students. The important part of this question, describe the sequence of events that result in an action potential. So if you know that the action potential should be made up of these 3 parts, depolarization, repolarization, and hyperpolarization. Run out of space on my slide. If you know the action potential is those 3 parts, then that's the important part. Depolarization, voltage-gated sodium channels open, sodium floods in. Repolarization, voltage-gated potassium channels open, potassium floods out. Bringing us back down, bringing us back down. Then we get to the bottom, hyperpolarization. Hyperpolarization, we have overshot that resting potential. We have gone into hyperpolarized, and therefore our sodium potassium pump needs to kick back in to bring us back up to the resting potential. So the important parts, and I will, for those of you who are still not sure on those intricate parts, please do go and look at my videos specifically on that, but I want to show you how this interlinks together. So if I've got a cell, and this cell is going to be connected to a sensory neuron, what's going to happen? So you've been told you've got calcium ions entering the cytoplasm of the receptor cell. Well, if calcium ions are entering, you should know calcium ions are involved in exocytosis. If exocytosis is going to happen, then vesicles containing a neurotransmitter, and they're not asking me about which neurotransmitter, they're checking, do I know that calcium causes these vesicles to release the neurotransmitter, and that will therefore bind to a receptor on the postsynaptic membrane. So my sensory neuron is my postsynaptic membrane, and that will therefore result in sodium coming in here. The sodium, the stimulus, gets up to the threshold value that therefore will trigger that action potential, depolarization, repolarization, hyperpolarization. So many students get confused because they start talking about the wrong bit. Describe the sequence of events that result in an action potential. So how is that going to cause this action potential? The neurotransmitters are being released from the cell. They will bind to a receptor, and that will therefore cause sodium to go in, and that will therefore cause that action potential to continue down. Guys, I know that you are going to be really cooked by this, but it's important that you note that it's so much to revise. I'm trying to give you some hints of how to answer questions that might not be there. Yes, Mia, that's exactly what it's asking. It's asking, do you know how it went from the receptor cell to the sensory neuron and triggers in that sensory neuron? So I'll leave this for you, but it wants you to talk about receptor, vesicles, neurotransmitters, binding, then sodium ions, then depolarization, and a threshold value being reached. So they're ensuring that you actually are able to apply that knowledge to that situation. Or they might do this to you. I feel this is the way they're going to go. They're either going to ask you to, why couldn't they just say that? Because that's not the point in the exam. The point in the exam is that you learning the content and then being able to apply it to the random situation. That's what they're saying. Oh, the first part of the question, that's because it was part of another part of a question. Because it actually, the first part in this exam was asking about evolution and how it evolved. We're not interested in that, and that's why. So if threshold value is reached, then depolarization occurs. Yes. So if the threshold value is reached, then an action potential will be passed on. It's an all or nothing. So depolarization is where you have the voltage gated sodium channels open. Sodium is going to flood in down its electrochemical gradient, and that's going to make it more positive and move upwards. Yes. What about, where have you gone? Oh, you redacted that just so I was reading it. Why doesn't the mark scheme talk about calcium channels opening? Because this is a different exam board. So this is a different exam board, but they've kind of already given you as the starting point the calcium channels, because calcium moves in. They told you in that question, if I go back up here, the first event in the sequence has been given for you. Calcium ions entered the cytoplasm. So they've already told you, or they've implied, the calcium channels were open. Calcium moved in. Therefore, that moves across. Causes depolarization of the membrane. Yes, Masood. If the threshold value is reached, an action potential is generated, that's going to result in that propagation of the impulse all the way down through the axon. So they can bring back some questions. Respiration, photosynthesis can come up anywhere, guys. Basically, it's interlinked with all of it. So you can't remove that from it. It's just how they ask the question. Oh, and it doesn't matter with OCRs one. Does not matter. Just work your way through. If you're AQA, then it's a completely different exam board, and I would start at the back. Right, when an impulse arrives at neuromuscular junction, so now what they could do is they could link the neuromuscular junction with the cholinergic synapse. So they could actually check your knowledge of a synapse with a neuromuscular junction. So do you know the similarities and differences between those two things? Do you understand that one is about a presynaptic membrane and a postsynaptic membrane? Well, hold on one second. Don't we have a presynaptic membrane and a postsynaptic membrane? Because this is just a neuromuscular junction. This is a synapse, but they're the same thing. They're gaps. Should really call this synaptic cleft rather than synapse. And this is the neuromuscular junction. So they both have presynaptic neurons. They both have acetylcholine. So this is my somatic nervous system, and therefore acetylcholine is my neurotransmitter. And we know acetylcholine is the one that is released at the synapse. Nothing. George, this is OCR A. There's nothing you need to know. Absolutely. So the sarcolemma, so this is the sarcolemma, which is basically a membrane. Well, hold on a second. Where does the acetylcholine have to bind to? Oh, yes, I remember. It has to bind to receptors on the postsynaptic membrane. So they must both have receptors for that neurotransmitter on that membrane. You don't need to know the blinking reflex. It was removed out of this one. It's just the knee-jerk reflex for you guys. Is there another one that's not acetylcholine, but it is not? Yes. So that would be if they're looking at parasympathetic versus sympathetic. So the noradrenaline is your sympathetic nervous system, but the somatic nervous system you should know is acetylcholine, and the cholinergic synapse you should know is acetylcholine. They're going to give you, yes, neuromuscular junction is between the motor neuron. This is my motor neuron, and this is my muscle, which will be my muscle fiber because we're looking at a cell. So if you're looking at the presynaptic and the postsynaptic, so this could be a sensory and a relay, then it's a synapse. What's a ganglion? A ganglion is a type of cell which is to do with the eye. It's not one that you need to know about for OCRA. So OCRA, you do not need to know about any of that stuff. Absolutely not. Don't worry about that. So what could you say? Acetylcholine, many mitochondria. Oh, why does it have many mitochondria? What's the important part? Oh, because some of you said about, will they bring up stuff again? They could bring up stuff again. If they bring up stuff again, then it's important to note that what they're looking at is, do you actually know that the mitochondria is for ATP production? ATP production because of aerobic respiration. So they could look at that. Yes, thank you, Renisha. You need ATP for exocytosis. For the cytoskeleton to move the vesicles, you therefore require ATP. So it's saying outline the similarity. So as you can see, it's literally just wanting you to state them. And this one gives you lots of different options. So therefore, this question I wanted to show you, because this is another thing they have done in a practice question before. And in the practice question, when they asked this, they didn't give it as nice a graph. So I quite like this graph. So if you are asked about a neuromuscular junction and you're asked about a motor neuron and the similarities and differences, it's important that you understand to take your time to dissect these graphs. So if they give you the graphs, then take the time. Where's the resting potential? Have you identified that they're at different points? Nice. There's no hyperpolarization. We can see there's hyperpolarization here, but there's none in the striated muscle fiber. What else can we see? We can see that the time period is completely different for the repolarization and depolarization. There's no refractory period. And also, if they've given you graphs, please, please use numbers, use data. -70, -90. This one is from 2 to 6, whereas this one is from 1 to 2.7 milliseconds. So actually give quoted data if you have a graph, okay? That is really important if they're going to ask you. And what they did on this question was they did exactly that. They asked the students to look at that data and then describe the differences. So they may do it solely from knowledge, or they may do it from a graph like this if they're going to do that. And they basically, I'll leave this in for you, just want you to talk about all the different points. Now, this one's a really interesting question. So do you have to mention everything in order, like depolarization to resting? I would look at the question, ask what they've asked me to see whether I should go all the way through. But if I'm talking about an action potential, I want to mention depolarization, repolarization, and hyperpolarization. TS, what do you mean you don't think you need to know if ATP is used? Oh, hold on, there's a question you answered above. Is ATP also used to reform? Yes. So ATP is required to move things back, but also it's also required for the adding together. Yes, you're absolutely right. But that would be a question. If they're going to ask that question, it will be linking to the fact that you've got the mitochondria, and they'll give you a bit of information on that one. Stop asking me that question. I'm ignoring it on purpose, and you keep asking it. It's not going to help me and make me do it any faster. I've got an idea of what I want to cover. I will look at things. You've got my other videos. Go and look at them. ATP is required to break cross bridges when we look at it with regards to the muscles, but we're not there yet. Okay, so looking at this then, there are 3 phases in a contraction of a striated muscle. So this makes, again, students freak out. They see a question like this and they're like, I don't know anything about these stages. Why have they given me this information? Well, they're going to ask you questions that are about you applying what you know to this situation, okay? So it's making sure you actually can take the knowledge that you have and apply it to a question. So with reference to this, explain what is happening in the striated muscle fiber during the latent phase. So what can we see here? We can see there's a stimulation, and then in this latent phase, what is actually happening? What are you going to have? If there's a stimulation, that stimulation is like the acetylcholine being released from the neuromuscular junction and binding to the receptor. So basically it's saying, what's next? What's happening after that has occurred? So it takes time for that to happen. The contraction phase isn't instantaneous. That neurotransmitter that has been released, acetylcholine, has to diffuse across. It has to bind to the receptors on the sarcolemma, and then we need the next step. We need the next part of this. So what's the next thing that needs to happen? We need the depolarization to move down the T tubules. What's that then going to cause? Well, if the depolarization moves down the T tubules, that is then going to result in my calcium ions being released. And if the calcium ions are going to be released, what will they then do? Well, those calcium ions are going to bind to the troponin. And what's going to happen when those calcium ions bind to the troponin? Well, when they bind to the troponin, they are therefore going to change its shape. When it changes its shape, what does that therefore do? Well, that moves the tropomyosin out of the way. So there's so much we could talk about. Now, if you are like me and you are dyslexic and you know your writing isn't the greatest, and you also know your spelling isn't the greatest, take the time to make sure tropomyosin, that you are spelling these keywords correctly, because they will penalize you if you are not putting those correct words in, and also making it so you are able to read your writing. It is annoying, but remember, these are not OCR. OCR will be much nicer than this, but I like the graph. I like the way that it's showing you. So if they give you a graph like this, it will be so much easier to read than what these ones are. How did I know, Anita? The reason I knew that, the reason I knew that that was what was happening next is because they told me there was a stimulation. The fact they'd already said there was a stimulation, therefore meant that I knew what's the next stages after. Oh, 1 second. What they could do is they could then link it to the relaxation phase and why there's a gradual decrease. So you can see this gradual decrease, and that links to your cross bridges breaking. So it's important that you remember that they're not going to ask it in the same way they would have done before. What I'm really interested in is this question, because I love the fact that they did this question. Again, this type of question, no OCR, just to be clear. I don't just do OCR, I do lots of other things, but it's good to do other questions because they will help you see where they could ask the question in the exam, okay? But just because there was nothing happening in the graph, what you have to realize, so Tanya, to go back to your statement there, which is a good point to make, it looks like nothing's happening, but you've got to remember the stimulation has occurred here. Something has to be happening. It just hasn't got to the point where you see tension in the muscle. And it's the same when you look at, for example, a population growth curve. When we talk about the lag phase, it's not that nothing's happening. It's that you aren't seeing a growth yet because they're getting used to the environment. So just because you have a straight line does not mean that nothing is happening. There's no tension in the muscle, but there are other things that are happening. And that's the bit that trips students up. And I'm not like other teachers, just to be aware of, I always teach holistically because this is how I see things. And this is how I see the exams going more and more, which is why students are like, it's so hard. And I'm like, well, just take the time and think about what it is that's happening. There's so much content, don't get me wrong. And after the summer, I'll have to recap it all. So I made my videos for me too, because I'll forget over the summer, just like you. Not as much, but there'll be things that I need to refresh and re-go over. Also, for the decrease of the graph, how does that relate to A to B being released? I'm going to come back to this in a second. It's really interesting. Because there's actually a question I want to show you. The next question kind of links to this. When neurotransmitters bind to receptors, does this cause an action potential to occur? No. When the neurotransmitters bind to the receptors, I've got another question to show you on that one too. When a neurotransmitter binds to a receptor, that therefore opens a channel. The ion, whichever channel that is, the ion is going to move in. That is either going to depolarize the membrane, and only if it reaches a threshold value will that therefore trigger that action potential. Be careful. Remember, the whole point of synapses is to filter out those low-level stimuli. So you don't automatically get an action potential in the postsynaptic membrane unless you have reached that threshold value. Yes, I do. I do have a video on muscle contraction. I've got a video on the whole course. Basically, it's all interlinked to playlists linked to your specification points. It's all there. Go and check it out. How long am I going to go? I said I would go probably till about 9 o'clock. I'll keep going through. I've got lots I want to talk through, but at the same time, I want to answer the questions that you guys have got. Oh, thank you. Do you know, I absolutely love it, but I also know how hard it is. This is the thing, and this is why I started my channel, and this is why I do this. I've only been doing this channel for, oh my gosh, actually, I missed it 2 days ago. My anniversary for this channel was 2 days ago. But yeah, I've only had this channel for a year. And I know so many people are like, I wish I found you earlier, but it's taken this long for people to get to know that I do these things. Right, coming back to this though, I don't want to miss out on this next question because this next question, it's a doozy, but I love it because it really is about applying your knowledge. So this question here is asking you, do you understand the hydrolysis of ATP during muscle contraction? So straight away, I'm going to take a step back and say to myself, right, what do I know? ATP is for detaching, breaking cross bridges between my myosin head and my actin. So therefore, ATP is about breaking those cross bridges between the myosin head and the actin. Right, I've got that. Now, hydrolysis of ATP. ATP can be hydrolyzed down to ADP and inorganic phosphate. Okay, but remember, this is a reaction that can actually go in both directions. Okay, so that's important for us to remember. ATP can be hydrolyzed down into ADP and inorganic phosphate, but we can also through, ooh, look at that, respiration, bring those two back together again. So that's something to think about. How do you know which ion channel is? You're going to have to look. I'm going to show you a question which says that, are you Claire Biology sister? No, I'm not. I've been watching your videos since October and you help. Oh, thank you. Thank you guys. I really do appreciate it. Do you start writing the answer before you finish reading? No, I don't. I take the time to give myself little notes, and then what I'm doing, I would obviously have more space. I'd write little notes above, but because of how I've got it on my slide, what I would be ensuring that I'm understanding is what are the hints they've given me. So they've given me words like hydrolysis of ATP, muscle contraction. So I know in my head, I'm going, right, what's ATP needed for in muscle contraction? Breaking the cross bridges. They've told me there's hydrolyzed down. Okay, ATP is hydrolyzed down. Well, I know we also need ATP. So I'm just going to show that that can go the opposite way through the process of respiration. So when you look at it as being a reversible reaction, well, it is a reversible reaction, but there's a whole load of steps that are required to do that reversing. I'm just triggering my brain to know where that's going to actually happen. Now, obviously, I need enzymes to catalyze the reaction. How do you stop yourself from regurgitating in the exam? The key thing is about regurgitating for the question. So I'm taking my knowledge, and I haven't answered the question yet. I'm taking my knowledge and I'm now going to look at what the question says next. So I'm just trying to trigger my brain, pulling that information. So when I was you and I sat in that exam, now I got a B. Just to let you guys all know this, I only did AS biology, and I got a grade B. It was a higher in Scotland. So I did year 12. I got a B for my biology, and I hated school. I hated learning. I didn't understand it. I wasn't very smart. And so I left. I didn't continue into year 13. I left with a higher, and that was it. I was done with education. I then worked in Tesco's as a red coat, is what it was used to be called in the old days. You had a red waistcoat that you wore. And I did a whole load of stuff in Tesco's for a year, and I thought, this is boring. I don't really want to do this. So then I went to college to do HNC and leisure management. Again, I did a year, didn't really enjoy it. I loved the biology, but not the stuff. So I only got a B in AS biology. Now I'm sitting here right now with a BSc honours in biology at university. I'm sitting here with a postgrad from Cambridge University for teaching science. I'm sitting here with a postgrad from Cambridge University on restorative approaches to conflicts in skill. And I'm sitting here with a master's from Cambridge as well. So I did not get a good grade in my biology. I got a B, which is a good grade. Just to be clear, when I say that I didn't get a good grade, I didn't get an A-level is what I mean. I got one higher, which was the biology, was the higher, which was my B. I got a B in 3 C's, English, maths, and PE. I got C's in. I got a B in 3 C's. But, I didn't understand, one, that I've got ADHD. So I didn't understand my brain works in a slightly different way. So for those of you who are like, why is she going off in tangents? That's why I do it, because that's who I am. But also, I didn't know that, and I only found that out 2 years ago, a year and a half ago. I only found that out. The second thing is, I didn't understand what learning was until I actually went to university and I was inspired by a teacher that I could actually achieve. And I applied to Cambridge as a joke. I applied as a joke, never thinking I would get accepted, and I did. Okay. I've then worked as a director of student progress for the well, I've been doing this for 2 years now, so the 17 years beforehand, I was a head of biology, I was a deputy head of lower skill, I was basically worked my way up until my girls came along, and now I do this. Okay, so yes, this is challenging, yes, this is going to be difficult for you, and whatever happens in August, just know this is not the end. If you don't do well in these exams, it's okay to take a gap here and try again. If you do well, amazing. It's the starting point of your next journey. If you'd have said to me, I've been making YouTube videos and doing this, I'd be laughing at you. I actually laughed when they said I was going to be a teacher, and look at me now. So it's really interesting that you are going to sit this exam and it's going to be challenging, but know that it is just a stepping stone. And it may be that it takes you a bit longer, but that's okay. I was a mature student equivalent when I went to university, and that's why I didn't need the A level, because I had life experience. I had other ways. I rocked on up to my Cambridge interview having no preparation done whatsoever because they didn't send me the email. So when I rocked on up and they said, so do your presentation, I went, what presentation? And then I just started talking. Obviously, I'm pretty good at that. I like the sound of my own voice. Anyway, back to this, right? So what are they actually telling us? Why is it important? Well, we have got calcium ions. Okay, so they're telling us we've got calcium ions. Well, we know we've got calcium ions. Calcium ions bind to the troponin. Troponin therefore moves the tropomyosin. Therefore, the cross bridges can form. So we know we've got calcium ions, but now they've given us something else. They've told us that these calcium ions are actually able to combine with the inorganic phosphate. So if the calcium ions are able to combine with inorganic phosphate in the sarcoplasmic reticulum, and this is going to form an insoluble calcium phosphate, and they're telling us that this may result in a fewer power strokes occurring in my sarcomere. They've basically told me lots of information that I'm not able to regurgitate, or am I? Well, what I am able to do is I'm able to say, right, well, if there's no calcium, it cannot bind to the troponin. Therefore, the tropomyosin cannot be moved out the way. If the tropomyosin cannot be moved out the way, the cross bridges can't be formed. So that's going to cause a problem. So therefore, that is going to result in a fewer power strokes. And I'll leave this for you to have a look at that mark scheme so you can actually see. Again, this is not your exam board, but it's the same content. And it's the same kind of style of question where they'll give you a little, but expect you to apply your knowledge to that situation. And then we can look at this one. Oh, actually, let me go back a second because Mr. Have I got another one? Somebody asked about the ATP, and you asked about the different less ATP can be regenerated. So yeah, absolutely. Something that was correct on your answer. Who mentioned about the ATP and the cross bridges? Oh, excuse me. Thing is, I always wonder how I lose my voice. It's because I just keep talking, right? I just want to show you one quick thing on. Let me add this in here. So just as a quick diagram of what it was, calcium binds the troponin, moving the tropomyosin out the way. The myosin head can now attach. Now it's important, you've got ADP and inorganic phosphate here. When they actually swivel, that's because they have released the ADP and inorganic phosphate. That is what triggers your power stroke. And that's obviously going to move your sarcomeres to be shortened because your Z lines are moving towards the M line. So that's important to note. And then what you have is your ATP will bind, and the minute the ATP binds, that's going to cause the breaking of the cross bridges. Now, this diagram here shows you that those ATPs are going to bind, changing the myosin's head shape and breaking the cross bridges. But if you have two of your myosin heads, for example, attached, and only ATP binds to this one will detach, this one's still attached. So they don't detach all together at the same time. So that's important to note, that they're not going to all detach exactly at the same time, because if they did, the muscle would just snap back. What you want to happen is this is kind of like it does this on each side, moving the Z lines towards the M line. I think the grade boundaries will probably be the same. Creatine phosphate. Creatine phosphate is just basically, excuse me, with regards to this one, the creatine phosphate is instead of it being the calcium phosphate, it'd be creatine phosphate, but creatine phosphate can give it back a lot easier. So creatine phosphate is like an intermediate that holds onto the phosphate to quickly transfer it back to the ADP to make ATP for more of the breaking of the cross bridges. So it's like your immediate storage molecule of ATP that can then be transferred, but it only lasts for the first 10 seconds. Then you need anaerobic respiration. Then you need aerobic respiration to kick in. Do I sell these slides? I haven't sold any of my YouTube live slides or any of my things because I'm making my booklets. I haven't got around to making the one for ATP, and you're so far to the end now. It wouldn't be worth it for you to buy these things. For the year 12s, they're going to be in luck because the minute I finish teaching you guys, all the time I'm going to gain back is going to be for finalizing each of the booklets so they've got it for the whole of next year. So I've got a few booklets on my website, ukbiotutor.com, but not any. So respiration's there, excretion's there, biodiversity is there, cellular control is there, and module 1 is there. But all the others I haven't finished yet. I'm working on them. I'll do my next live again tomorrow. So that it helps. Yeah, but you've got to remember the grade boundaries are going to be changed depending on one, how hard the exams are, and the cohort that sits them. And that's the thing you've got to remember. Every grade boundary changes depending on the cohort, and you don't know you're going to retake yet. So, but great, go buy my booklets, absolutely. So the creatine phosphate side of things, hold on, I'm not finished on this. Let me just, where's my muscle? So next thing to know is ATP hydrolysis, that breaking it is going to provide the energy to open up, and therefore that can reattach. So the creatine phosphate is basically just the fact that you've got ADP can become ATP by taking the phosphate away from the creatine phosphate. Creatine can take the phosphate and store it. ATP cannot be stored. ATP is used and it is broken down because that third phosphate is actually repelled by the second phosphate and the first phosphate. So the molecule is a much more stable molecule as ADP. And so therefore, because of that, that's therefore going to break away. And so creatine phosphate is a way of storing it to quickly transfer it back by substrate level phosphorylation. And that therefore helps. These questions are a mix of exam boards. So some of them are CIE, International A-Level. Some of them are EdXLB. Some of them are AQA. Some of them are OCR. But the ones I'm showing tonight, none of them are OCR, although the content and the questions that I am showing you are relevant to your content. So the reason I've picked the questions that I've picked, I spent a whole load of hours researching and planning and organizing this because I want to make sure that I don't teach you anything that's unnecessary. Okay, everything that I'm giving you is your content specific and questions that could relate to your content specific side of things. What happens to the calcium phosphate? Well, just like anything else, you would need to have a way to break it down. So therefore, it would be that there would be an enzyme that would catalyze a reaction to break them down, or it would be a case of it moving in another way. So it's important that you note that you're not expected to memorize these bits of information. So all the things that I'm giving to you now, you're not meant to memorize the questions. This is where they're expecting you to memorize that content and it coming through. And fermentation I did in my last live. So if you look at my recorded videos and my live streams that I've done, it wasn't the last one, it was the one before, but I absolutely I've done that bit already. I want to make sure that I've covered enough content that I haven't done in a while. So in this one, explain the need for many mitochondria in a presynaptic knob and sarcomere. They want you to note that what the mitochondria does. So again, that's linking back to module 2 and then moving on to modules 5. For production, for making, active transport, detaching, and also your sodium potassium pump. So don't forget about that. And I've got one more link to neurons I want to do, and then I want to move on to genetic engineering for tonight, because I want to make sure you guys have covered this. Ooh. Okay, so what have we got here? We've got a question that tells us lots of information. So this is where they could ask you to apply your knowledge. They love throwing in a new neurotransmitter that you haven't heard of before. So you're told in the question, the stem of the question, that acetylcholine causes the influx of sodium, glutamate, influx of chloride ions. So basically, we've got our pre, we've got our post, we have 2 neurons. Those 2 neurons both have vesicles. Now, one of those vesicles contains acetylcholine, the other vesicle contains glutamate. So therefore, that must mean that there are channels that are specific to those neurotransmitters. Well done, spatial summation, absolutely. And therefore, you know that if those neurotransmitters are going to be released by exocytosis, they are going to bind, open the channels, and you've been told this channel lets calcium in, and this channel, oh, that's not calcium. Why am I writing calcium? Chloride even, in, and this one is going to let sodium in. So therefore, Na plus, and this one is Cl minus. So it's checking, do I know my ions? So let's just take a step back and remind ourselves about our resting potential. If I've got my resting potential, if neurotransmitter X is released, that is going to take it in one direction. If my neurotransmitter Y is released, that is going to take it in an opposite direction, because this is a negative ion. This is a positive ion. And remember, we're at a membrane potential of 70 millivolts. So it's going to take it more negative and it's going to take it more positive, depending on which one goes in. Would this question follow up with inhibitory postsynaptic potentials in the excitatory? Well, let's have a look at what this question did. Because I love this question again. And that's why I tend to show you guys is questions that are, again, very similar to what they could do. So this one caused depolarization, and this one caused hyperpolarization of that postsynaptic membrane. So then they said, okay, so what do we have? We have our students, and those students are going to make a conclusion. Do you know which way round it goes? So let me just bring this up here. So what have we actually got? Well, when only neuron X is stimulated, an action potential will occur in the postsynaptic neuron. Well, we know that that was depolarizing. So therefore, we can say, yep, we do expect that one to happen. When only neuron Y is stimulated, an action potential will occur. Well, we know that Y actually caused hyperpolarization. So therefore, we would not expect that to happen because we're going in the opposite direction. And then the last one, when neuron X and neuron Y are stimulated, an action potential will not occur. Absolutely. That is true. So when it says explain whether or not you agree, they won't say that for your exam. They'll say evaluate the student's statements. Exact same thing. Agree with 1 and 3, disagree with 2. So whenever you get a question like this, I love these types of questions because what they're actually doing is they're checking your knowledge of what's going on. And so therefore it will link to your content. Right. I'm going to jump around now. Oh, one last thing to just bring in. Multiple sclerosis, they've asked questions about that in the past. Multiple sclerosis is going to affect the myelin sheath. Myelin sheath being broken down. Remember, myelinated axon, you've got your Schwann cell. Your Schwann cell is going to put up to 20 layers of membrane, phospholipid bilayer going all the way around. Why don't you understand? Hold on, this one. If I've got one that's causing, where am I on this one? If I've got my resting potential and I have got one that's depolarizing, but one that's also repolarizing at the same, hyperpolarizing at the same time, it's going to cancel each other out and therefore keep me at my resting potential. So neuron Y cause the neurotransmitter to open the chloride channels. The chloride channels lets chloride going in. And because chloride ion is a negative ion, that is going to prevent it from reaching that threshold value and therefore ensures it stays towards that resting potential. Right, that's a good one, Anita. Good question. If X is stimulated more than Y, then you could say as long as the threshold value was reached, then an action potential would be triggered. So you would need to have enough of a stimulus, enough of a depolarization for that to occur. Yes, it could show graphs. Absolutely. Yes, it could, and then expect you to apply that. Why doesn't hyperpolarization cause an action potential? Because it's taking it too far away from that threshold value. Remember, an action potential is depolarization, repolarization, and then hyperpolarization. So the key thing to note is if you are coming away from that threshold value, if you're becoming more and more negative inside your inside your axon, And it actually links quite nicely, which I'm about to go on to. It links to the release of the insulin for glucose as well, which is what I'm going to talk about. Okay, moving on. Yes, I am going to do another live tomorrow, but I won't cover any of these topic areas tomorrow. I will move on. Exactly. This is the thing. This paper is so unpredictable, which is why I've looked at what I think would potentially come up, and I'm trying to show you ways in which it all interlinks. We wouldn't say that, we wouldn't call it neutralization. We would just say that they cancel each other out, which is where you're trying to go with neutralization. But we just say they would cancel each other out. What have I covered? Lipids, membranes, muscles, synapses, neurons. I'm about to go on to hormonal communication now. I think I don't know. I need to have a look at it to know for sure. But the myelin sheath, I want to bring you back and look back around to the fact of that lipid layer and that insulating layer of the myelin sheath. They could link those together. I do feel like lipids are coming up. Basically, anything to do with biology is either proteins or membranes. Yes, I did. I did literally hand make every single one of these slides, and every single one of these diagrams is also mine. That's what I was learning at the start of last year. I spent actually, no, hold on. I started learning how to use Illustrator first when I was still teaching in the school that I was teaching, so I could make the diagrams better. And I'm still learning. I'm not fully there, but I'm still learning how to do them, but I'm getting better with each one. But yeah, this is why, and that's why my workbooks are mine, which take me time to do. So Schwann cells are cells. Remember, they are differentiated cells, cells which have then dropped a phospholipid and wrapped themselves around. Every single cell that has differentiated for the role that it has to play, whatever that role may be. Okay. And so therefore, if you understand that Schwann cell is a cell that has a membrane, got your nucleus, that's how you know that's the Schwann cell. That Schwann cell has then wrapped itself around and around and around, is basically created a big massive layer surrounding and blocking off the sodium potassium channels. No, it's all interlinked. So yes, they are specialized cells that have got a specific role to play. That role is to wrap themselves around, creating the myelin sheath, which is the insulating layer of the neuron. And that neuron is in the peripheral nervous system, which is part of your, yes, so they could link it to stem cells. I've got a question, actually, I'm going to link to stem cells if we actually get onto it. I've got another hour at least to go, so I will. Is it stem cells will come up in lots of different ways. It just depends how they come up. Right, this is what I want to go on to now. So let's move on. We're going to now look at, correct me if I'm wrong, nothing came on cyclic AMP. Yes. Mia, yes, they do. Oh, excuse me. As gross as it is, I am moving things around that's making me, do you need to know something about, no, not for OCR A. No, you do not. But stem cells came up on paper too. They did, but I haven't seen the paper, so I don't know how much stem cells came up in paper two. But just because something has come up in paper two doesn't mean it won't come up again. What it's saying to you is it's only going to, so if it came up as a multiple choice, let's say, it therefore could come up as a question where you're applying the knowledge. Yeah, exactly. So I'm saying there's nothing more that's come up. They could have brought in cyclic AMP linking to paper 2 and linking to post-translational modification, which is why I want to double check nothing came up on that. It's like a 2 or 4 marker. So yeah, it just depends where they want to go. But I'm going to show you a question which actually links to gene therapy in a minute. I'll say in a minute, probably about 20 minutes. But let's look at this one. So here we're told we've got adrenaline. Adrenaline binds to a receptor and it activates a G protein. Now, you don't need to know about the G protein for your exam board, but you do need to know about A, which is why I like this question. So in this case, they expect you to be able to identify what A is pointing to. So therefore, you should know about adenylene cyclase. And you should be able to label that because there is, in the picture, ATP being converted into cyclic AMP. So you should know already that this is your primary messenger, and then cyclic AMP is your secondary messenger. And I believe they haven't covered really anything on this. No. Adenylene cyclase is the enzyme that's going to be activated to produce the cyclic AMP, which is the secondary messenger. So if you were looking at your adenine attached to, because again, they haven't really done anything on nucleotides and nucleic acids, so this could come in. If we're looking at cyclic MP, we've got an adenine nitrogenous base, we have a ribose sugar. We know it's a ribose sugar because it's got a hydroxyl group at carbon number 2, just so you guys remember. And it's got 1 phosphate. So this is a nucleotide. That is a nucleotide. Now, you know ATP is basically adenosine triphosphate. It's exactly the same. It's an adenine, it's a ribose, but this time I've got 3 phosphates. So I should know the structure of cyclic AMP because it is a monophosphate. It's adenylene with 2 YLs. I'm not just, that's one thing. You have to know how to spell it. And it is 2 YLs. So it's important that you write it in the right way. This is hormonal communication or it's animal response. It just depends where they want to do. Yes, it's always within the membrane. Remember we talked about the fluid mosaic model and the fact that it's a mosaic. So it has these proteins, these intrinsic proteins, and those intrinsic proteins which have the capacity to move around. Well, the G protein is a peripheral protein which has the capacity to move around as well. And if they ask you to label it like they're doing here, they would say label A and B. So yes, they could. Are these short lives condensed revision? This is not a short live. This has been 98 minutes so far. But it is basically a revision of the topic areas. Can't believe I've been going for that long. But yeah, and consistently had over 100 people watching. This is amazing. Thank you, guys. Yes, it's just a transmembrane protein. Absolutely. That's all it is. So the question though on this one is asking about B. So adenylene, be careful, 2 YLs. So adenylene, YL, cyclase is an enzyme which catalyzes the reaction of ATP to cyclic AMP, and therefore a secondary messenger is produced. So the adenylene cyclase is the enzyme. Look, it's got A's at the end. So that's what tells us it's an enzyme. Yes, YLYL. Oh, which textbook are you using? Hold on, let me just double check that. Let me double check if I can see if there's a question on it where they've disallowed. Okay, so I'm just going to show you, and this is specifically from the exam that was written. This is a 2023 exam. So this was one of the questions that came up in the 2023 exam. So what I would say, which is where I get all my resources from going through the exam board specifically, look at the spelling. So just be careful. You're not going to lose a marking point because you haven't spelt it the right way. So I don't know which book. So hold on, guys. I don't know. Official one. If it's in the official OCR A one, so it's definitely not in the black one that I've not got it here. It's in my other inside. The CGP guide says the same. Interesting. Okay, so if it's in an endorsed OCR Pearson's textbook and it's been endorsed, textbooks can be endorsed by OCR, revision guides can't, so they're not endorsed. But if it's in an actual endorsed OCR Pearson's textbook, then they will accept it as both. Right, let me go back to the question. Hold on 1 second. So yeah, just be aware of that one. So it is definitely adrenaline. Not long ago saying, right, okay. Oh, thanks for that. That's what I thought. I can't find the question. I did believe. Yeah, but is it an endorsed? How long ago was it endorsed? Like, what was the date of the publication? If it was a publication which was before 2017, then that would be one where I would say that it's not been updated. So therefore your school may have given you an old textbook. It is definitely safer to put adrenaline. So this was the one that I just showed you a minute ago was 2023 straight from the exam paper. That is what they're expecting you to do. That is what they're expecting you to write. Now, I'm not going to lie. I had never used adrenaline cyclase before in any of my resources when I was teaching this because I'd used the old style. There we go. It's not updated. So that would be, do not write that. Please, 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 which is why I had to ask the question. You need to be using up-to-date resources. So all my videos that I've got out there are up to date for the 2025 exam that you're sitting right now. So I only started this YouTube, as I've said, and I won't go down that pathway, but I only did it last year. So I made sure that all these resources are very specific to the content that you need to know for this exam. What's the difference between the two? Good question. Don't know. Let's move on. But OCR wanted this way, so we're going to go down this way. It'll be a case of that the fact that it's actually the enzyme's got the different name. But I don't know. I'm not even going to try and make it up. I don't know, to be honest. My brain can't work. I've been going at this 104 minutes. And I want to just keep going to finish what I want to get done tonight. So yeah, not going to lie. I don't know it all. I don't. I know bits of it. I have prepped for this session. I have made sure I've got what I needed for you guys. And that's what your teacher does. Your teacher goes with the resources they have. And unfortunately, your teachers also have to teach so many other topic areas. I teach lots of different exam boards, but I have made my resources and I literally know OCR inside out, but I still have to double check. And yeah, genetic engineering is coming, I promise you, because that's the other one I think that they would go down the route of. But this one, what are they? They're asking what B is. You should know that B is a protein kinase. So you should know that B is a protein kinase, and therefore know that they could ask that on that topic area. So what I want to now look at is I'm going to link quickly to the pancreas before I go on to genetic engineering, because it all interlinks. This is the thing about biology. Biology is they can go any which way they want and interlink it to module 5 and 6. So anyway, here we go. You should know, up here, I've got my beta cell, and you should know that if I've got a high glucose concentration, that glucose concentration is going to be broken down by glycolysis. Then I've got my respiration occurring, aerobic respiration inside my mitochondria, and that's going to produce my ATP. You should know that I've got my potassium ATP channel, and my potassium ATP channel is going to close if there is ATP being made. So when there's high glucose, that is going to result in more ATP. More ATP is going to close my potassium ATP channel. So here I have my sodium potassium pump, whether I'm talking about neuronal communication or whether I'm talking about this, the same. 3 sodium out, 2 potassium in. Therefore, I have a high potassium concentration, which should leave through an open potassium channel, but I've closed it. So that potassium's not leaving. So because that potassium's not leaving, the membrane is going to become depolarized. And because it is depolarized, that is going to result in my calcium channels, my voltage-gated calcium channels opening. Calcium is flooding in. Calcium is therefore going to cause my vesicles containing my insulin to fuse with the membrane. Whether I'm talking about synapses, whether I'm talking about, this case, insulin, a hormone being secreted, I'm actually talking about cell biological membranes and transport across the membranes. I'm talking about movement with the cytoskeleton. So I'm talking about module 2. And this is where it's all interlinked. And this is what you have to understand. So a high blood glucose level is going to be detected by my beta cells, going to result in my insulin being secreted. The insulin can then be detected by the target cells, virtually every cell around. So therefore, what we've got is the insulin binds to glycoproteins. Let me think this one through. We're talking about a phospholipid bilayer. We're talking about glycoproteins. We're talking about these receptors that are going to be on the surface of the cell. That means the insulin can bind to it. Therefore, what's going to happen? They're going to change their tertiary structure. When they change their tertiary structure, insulin can move in. We are now decreasing the blood glucose levels. Insulin is going to either increase the rate of photosynthesis. So if we've got more glucose going in, so the insulin binds the receptor, the glucose is taken in, therefore the insulin has signaled for an increase in the rate of photosynthesis. Oh, it's not. Oh my gosh, I'm getting tired. The rate of respiration. Do not do that in your exam. So insulin is going to result in an increase in the rate of respiration in the cells, not photosynthesis, respiration. What else could happen? Glycogenesis. What is glycogen? Glycogen is a polysaccharide biological molecules. It is a molecule which is branched, a molecule that is branched because it has 1 to 4 and 1 to 6 glycosidic bonds. So the glucose is going to be added through a condensation reaction. The glucose is going to be added at the ends, and therefore the insulin has signaled for the activation of the enzymes inside. Yes, always jump to tertiary structure. The primary structure would be if you're looking at a mutation that changes, therefore the tertiary structure. But absolutely, every single thing you should be going, unless you're told that it doesn't have a globular structure, then if it's not got a globular structure, it's not the tertiary structure. The links are crazy, and that's what basically I'm trying to help you to see. Yeah, exactly. AQA do the essay because it all interlinks. You guys just don't get it in OCR as a big 25 marker. You get it as a, here's the information, dissect and pull it out. So this is for the reason OCR exists is because some students can't deal with a 25 marker essay. So instead it's given to you in the way of the questions. Probably about the same. I just need to double check what time I can come out, but probably I'll go live about 6:30 again tomorrow. I have no idea what that person asked. Where are you? Guys, I'm trying to keep up with what you're asking, what you're putting in here, but I have no idea. Keeping going, lipogenesis. Does glycogenolysis and glycogenesis relate to fight or flight response? So glycogenolysis is the breaking down. So that would be where you're looking at your fight or flight response. So what do you want to do? Do you want to build the glycogen up or do you want to break it down? Is there more application in paper 3? It depends all the way through. They have a set percentage they'll do as a maximum in the exam. It just depends on that one. So the fight or flight, but glycogenolysis, yes. What's the difference between adrenaline and noradrenaline? Adrenaline and noradrenaline are just different structured chemicals. Noradrenaline is also a neurotransmitter. Should we probably recap what's come up? It's a hard one. I don't want to say no, because if I say no and you don't do it and they ask a question on that, then you might go, oh, she said no. I don't think they'll go into as much detail, but again, it depends how they've asked the question. All the stuff on respiration, interestingly, was more about enzymes and your knowledge of enzymes. You had a couple of marking points in there, which were like, you had to name the link reaction, the Krebs cycle, for example, which was AO1. But with regards to the other parts of the question, it was all about enzymes and inhibitors of enzymes and enzyme actions rather than actually about respiration. So do I feel like respiration is coming in again? Yeah, I absolutely do, because it can link to so many things. It can link to the carbon cycle. It can link to basically anything, because every cell needs has a metabolic demand and therefore needs ATP. So it links in so many different ways. I definitely think do the stuff that you are not confident with. So this is us all talking about the liver and insulin that was produced from the pancreas, and obviously glucagon, and the fact that if low glucose concentration is detected, this time by alpha cells, It's going to respond by producing glucagon, and I always go a glucose, agon. So glucagon is the chemical messenger that's going to bind and say glucose needs to get out of here. We need to raise the glucose back up, and so therefore glucagon is going to be the hormone that's going to go to the liver cells, and that's going to therefore signal for the liver cells to release the glucose. So when the glucagon binds to receptors, it's going to cause the glycogen, it's the opposite, glycogen to be broken down into glucose. The glucose is going to be released. So therefore raising it back up. I definitely think there's going to be a planning investigation because I haven't done that yet. And you've got gluconeogenesis, where you've got your glucose and your amino acids, which can be converted into glucose to bring it down to increase your glucose molecules. Amna, good to see you. Amna, I've been missing you in my sessions. And then you've got negative feedback. Obviously, what is negative feedback? You're going to have this whole thing about bringing it back up to normal. So in this case, insulin and glucagon are a really good example of negative feedback. And the reason they're a good example of negative feedback, if my blood sugar levels get too high, insulin is going to bring them back down. If my blood sugar levels get too low, glucagon is going to bring them back up again. Right. Where am I going on? I don't even know where I am anymore. Where am I? Where am I? Where am I? I'm on that one. Yes. Okay. Right. I want to show you a question again where they could ask you some application. Are we required to know the practical on animal movement? I just saw a video saying it's not been asked for OCR, so it may come up. Animal movement is, when they're talking about that, they're probably talking about animal response and hold on, right. So animal movement, if they're asking about animal movement, they're asking about animal response. So it would be more about muscles. It's not about chemotaxis or anything like that, because that's not part of your course. But I'm intrigued of what they meant with the practical and animal movement. It would be animal response, how an animal responds to something. It would be about woodlice, apparently. Well, the only thing you could do about woodlice would be looking at how it responds to changes within the environment, but you wouldn't really do that. But if they are going to do it, they would give you something on it. They tend to do caffeine and heart rate on organisms, but then they look at the ethical side of things. You shouldn't really do that. But that would more be Daphnia than it would be about woodlouse. So make sure you're looking at a video that's definitely OCR based, would be what I say. I don't know what they're mentioning when they talk about that. If you're talking about whether it's an endotherm or ectotherm, then that could be something they look at before, but I saw a video saying it was PAG 10. Yeah, PAG 10 is animal. Let me just double check that. No, PAG 10 is about what do you call it? It's your visualization and your OS2. Let me just double check this. Hold on 1 second. Because I don't want to have wrong information. So let me just double check. PAG 10 could be data loggers. Hold on, I'm going to the wrong place. I've got too many files. Too many files, too many files, too many files. Where are you? Where are you? Are you? Sources from here. Here we go. Investigation, yeah, using a data logger or computer modeling. So if they're going to give you something on that one, they will give you an information about this. So for example, if I'm looking at the ones, you can either have, I did DNA Rasmol with my students. You can do turbidity of a microbial broth increases as the culture grows and density of cells increase. So you could have done that. Or you could have done, the other example is measuring pH changes during yogurt production. But that doesn't necessarily mean that that has to be done. That's the thing about OCR. There is no set practicals with OCR. It's very much based on what it is they're assessing. So PAG 10 is investigation using a data logger or computer modeling. And so that's what they could do. So if they're doing anything to do with animals, you'd be looking at, like they did one once, which was about a fish using an oxygen probe in the tank, looking at how the oxygen was being used. It is really hard to tell what's coming up and what's not. Did culturing microbes come up in paper two? I don't believe it did. So I do believe that that potential is coming up. And I said before, one of my educated guesses would be that you're going to have to do yeast respiration in some form of culturing of it for moving it to a bioreactor, would be potentially where. Yeah, it's one of those things where there are lots of different people who are making lots of different videos, and some of those videos are amazing, some of those videos are good, some of those videos are not so good. What I would say to you is always refer back to your specification, making sure that it links to it, and knowing that different schools can do different things. But PAG 10 is about you using a data logger or a computer modeling system. So it really just depends on what that teacher decided to do. So they may have done a video based on a practical that they decided to do, which was using a data logger. And therefore, if they have, that's what they're trying to talk to you about. But make sure, because the animal response one is actually PAG 11. So PAG 10 is just about your use of a data logger or computer modeling, and it can be done in any which way they want to. Yes, they could ask you about temperature, different temperatures on the rate of anaerobic respiration, because you'd be expecting different kinetic energy, and therefore different kinetic energy, different rates of reaction. So that could definitely go on that one. On this question, just to show, this one was looking at you understand the effects of the rate of glycogenolysis. Glycogenolysis would be to split glycogen. If the blood glucose concentration was low, we would expect an increase in glycogenolysis, a decrease in glycogenesis, glycolysis. Because my blood sugar levels have decreased, I would expect a decrease in glycolysis. And if my blood sugar levels are low, I'm not going to want to convert them to fatty acids, because that's a way of storage, and therefore decrease. Yeah, don't go through all the content. When is cyclic AMP involved? Well, in this case, what you'd have is glucagon would activate. In this case, they had to know about the G protein, you don't. It would activate the adenylate cyclase. The adenylate cyclase is going to activate the ATP being converted to cyclic AMP. Therefore, you've got an enzyme cascade. Now, if we're going to split up glycogen, we're going to increase the rate through using enzymes. So Hold on 1 second. Oh, I'm gonna lose my voice. Where is it? It's in my old stuff. Week 6. Yep. 1 second. I'll find you a picture. My really old. This is going way back. Where are you? Where are you? Where are you? Here. Just gonna throw it in a slide here. So here you have your role of your liver, storage of glycogen. Obviously, the insulin binds the receptor. That's going to cause a cascade of events. So increase the permeability, increase enzyme action. So that's an interesting one, actually, because I haven't talked about that. If insulin is going to increase the membrane permeability and enzyme action, therefore, it's going to increase the inputting of a protein. And so therefore, do not think that you are all failing. That is not true. What you have to understand is that it's going to be tough. To be honest, one of the bit always, I find paper 3 easier, but that's because my brain works in that sort of way. But I would say paper 3 is going to be a mix of, there'll be some easy questions and some hard questions. But I feel like if you were to rank them so far, paper 1, I think, has been the hardest. Paper 2, I think for most people, maybe I'm wrong on this one, but most people are feeling that paper 2 has actually been a little bit easier and they've been able to apply their knowledge to paper 2, which makes me think paper 3 is going to be in between. It's going to be a bit of a mix of the two. Oh my gosh. So yeah, protein kinase basically activate lots of different things. No, lipogenesis, genesis means to make. Lipogenesis would be to make lipids. So this would be if I've got too much glucose, if my blood sugar levels are too high, what I'm going to do, I'm going to remove the glucose, store it as glycogen, or convert it into lipids. And that would be where your lipogenesis comes in, or lipogenesis. Opposite way around, gluconeogenesis is where you're going to create glucose from the lipids. Gluconeogenesis. Genesis means to make, neo means new. We're going to make new glucose. Glucose from another molecule. No, it doesn't just apply to lipids. You're absolutely right. And it also applies to proteins. If you were asked a 6 mark on how to plan the investigation, you're going to trip yourself up. But the key thing I would say is IV, DV, CV. What's your independent variable? What's your dependent variable? That's normally in the starting part of the question. Then what are you going to control? Have all your volumes. If you're not sure, definitely check out my module 1 booklet. I don't know if anyone's on here who's got my booklets, but if anyone has and thinks it's helpful, please do let us know for other people. But I would say I went through how to do the different practicals, but I didn't do specifics because I don't know where they're going to go. So having your list of variables and then just applying them to the situation. So IV, DV, CV, stats test, standard deviation, absolutely. Repeating for means, there we go. W's got it. You think there will be a lot of application paper? No. What you have to realize, and I keep going back and saying this to you all, that there is a thing that you should be doing, or let me just get the specification up. It's towards the end. On your specification, oh, where is it gone? Where is it gone? Just do a quick. I take it. Right, so this is what AO1, AO2, and AO3 actually refers to. So this is on page 53 of your specification. And then do this. So AO1 is demonstrating your knowledge and understanding of scientific ideas. That's basically recall, right? Demonstrate your knowledge, process, technique, and procedures. AO2 is apply that knowledge and understanding to scientific ideas, processes, techniques, and procedures in a theoretical context, in a practical context, when handling the data, both qualitative and quantitative. And then AO3 is about, here's a set of data, here's a bit of information, here's the stem of the question. Can you now analyze, interpret, and evaluate what you've been given? Can you refine, give improvements to practicals? Can you design practicals? So then if we look here, paper 1, paper 2, paper 3. AO1 is anything 13 to 14% in paper 1, 13 to 14% in paper 2, 5 to 6% in paper 3. So therefore, in total, can be anything from 31 to 34%. So a very small amount of it in each of the papers is recall, and that's what a lot of students don't understand. So when you look at this, they therefore are going to be lowest 31, 40, 25 potential, but it's all mixed because obviously you're going to get to that 100%. So if they do more AO2 in one paper, there'll be less in another, but the least it'll be will be down to 10. No, they don't give you, because it's all interlinked. Abigail, all of it is interlinked. It doesn't matter where you go. If I said to you, give me a topic area, tell me what you think the topic area is, and I'll find you a link with other areas. All of it is interlinked. Oh, hold on. Amna's giving me information 61. Maybe it does. Maybe I'm wrong. Amna, am I wrong? But on 61 shows me the how science works part of it. So are you meaning on, so 61. Oh, for the percentages. No, that wasn't 61. That's on 53. Okay, that's what I was about to say. What? Where's that? I missed that. Okay. So increase, decrease. Oh, this one is one that I really like. Oh, talking about AO3, for example. If they do go on to talk about genetics, which I'm about to now lead on to, by the way, guys. So I want to do a bit on genetic engineering, but not the way you think I'm going to give you information. It's because I've got the more updated one. That's why I'm using now the updated specification. So it could have been on the old one on page 61. Yeah, Emanuela, I'm just about to do that now. That's literally where I'm moving on to, because that's where I'm going to finish on genetic engineering. But I'm going to ask it in a very different way, or show you in a very different way. So I extracted DNA, or at least I tried to extract DNA from strawberry plants. So they could give you a method like this. Take a fruit from a strawberry plant and crush it using a pestle and mortar. Add salt to the crushed strawberry fruit mixture, add an enzyme to the mixture, add ethanol to the mixture. So they're checking, do you actually know what you should be doing and why you should be doing it? So do you know that you want to grind the sample up so that you're breaking the cell wall apart? So if I'm going to do genetic engineering, I need to isolate the gene. Well, I need to isolate the gene from the DNA. I need to get the DNA out of the cell. I need to get the DNA out of the cell's nucleus. So I need to crush it up first. I need to grind it to break the cell wall. I then need to use detergent to, oh, look at that. Break down plasmas. Let me think about detergents. Are detergents non-polar or polar molecules? Because they are hydrophobic tails, they're going to insert themselves into the lipid bilayer, disrupting the structure. Biological membranes coming through again. I'm going to use salt. Salt's going to neutralize the charge of the DNA. It's going to neutralize that sugar phosphate backbone, making it less soluble in water for that precipitating out. I'm going to use a protease enzyme to break down the histone protein. And then I'm going to use alcohol, which is a non-polar molecule. So that's going to separate the DNA out to form a precipitate. So what do I need to know? Well, DNA inside the strawberry plant is a eukaryotic plant, therefore it's inside a nucleus. It's also got histone proteins. So I need to break those histone proteins away so I can isolate the membrane. Yes, it will always disrupt the membrane. It's basically what they want to assess whether you can do or can't do. And that's why you get all the way from A star down to E grade, because it's about how much are you able to do this. That's what they want you to be able to do. They're going to give you hints in the question. That's the key thing to note. They will give you the hints in the question. It's important that you know how to pull that information out of the question and then add in a little bit more of what you've got. You can do. So, Mia, if they were asking you about the water bath, what would that be for? Why would a water bath be important? And I swear you think like an examiner now. It's great. I love it. But it's an interesting one. If they did say the student were to heat it, why would they heat it? Why is a membrane disrupted and destroyed? Absolutely right. Just table salt is absolutely fine. Remember, the minute you put table salt into a solution, what you're going to have is you've got your hydration. Your hydration. Hi, Millie. The hydration thing. Millie, you need to watch the beginning of the video. I've gone over all the neuronal stuff at the beginning. So Millie, please do check that out on the video. You need to be like Amna. Amna has been working solidly, and Amna's come to every one of my lives since I started. But Amna has done so much work. It is impressive. Oh, interesting. Well, but Mia, going back, if your teacher did it with a water bath, why did you do a water bath? A water bath would control the temperature. So it could be that you're looking at a control variable. So if I'm using a water bath, I'm controlling the temperature. Therefore, I'm ensuring there's nothing else that's involved that's going to affect my practical. The other thing to note is if I'm increasing the temperature, I'm increasing the kinetic energy, I'm therefore disrupting the membrane more, because remember, if I've got an increase in temperature, I'm going to disrupt the membrane, therefore allowing it to expose what was there. So just showing here, take the fruit, crush it. Why? Breaks down the cell wall. They didn't mention about a detergent. That's not a good thing. Add salt. Why are you adding salt? Breaking hydrogen bonds between the DNA and water molecules, making DNA less soluble. Add an enzyme to the mixture. Obviously, they didn't mention it was a histone enzyme, and then the ethanol precipitate out, disrupt the cell membrane, and it needs to be really cold for it to be reduced. So what I want to look at here is now linking it to genetic engineering. Ideally, you should be specific with the order, but if not, it won't matter too much apart from if you were to do like a step the wrong way. Well, it would matter if you try to do something in the wrong order. So yes, actually, it would. Yeah, in this case, if it doesn't matter with regards to different orders in certain things, but if there are things that are specific orders, then yes, there would be an expectation. Okay, stop distracting me, guys. Stop talking blesses about this. Okay. Cool, what we at? 11 minutes until I said I would finish, but here we go. Right, I'll probably go a little bit longer because I want to cover this genetic engineering side of things. So again, this is back to questions that are not from OCR, but it is very much linked to where they could go. So this one is talking about a enzyme you've never heard of before, adenosine deaminase. You've never heard of that before. It's a deficiency in the immune system. So they haven't done much on the immune system, so they might bring it in a different way. And the fact that it's a recessive autosomal mutation, because I don't believe, unless I'm wrong, that they did much on mutation. So if I'm wrong on that one, please correct me, apart from mutations causing variation, etc. But they could bring in a question which just mentions it, and therefore if it mentions it, it's their way of saying, oh, tick that box, we've talked about mutation in this paper. So then you're told you can have severe combined immunodeficiency caused by a lack of this ADA, and therefore your call is called ADA SCID. Genetic engineering is used to make a recombinant human protein to treat people with ADA SCID. What's an autosome? An autosome is chromosomes 1 to 22, which are made up of homologous chromosomes because they have the same genes at the same loci. So the same genes at the same location. Yeah, I didn't think there was much on mutation. What's a recombinant human protein? Right. So recombinant means different from the original. So crumbs. So different from the original is a recombinant. Human protein is a protein that's found in humans. So if they go like a question like this, outline the principles, they didn't need any of the stuff above. They really just wanted you to outline the principles of genetic engineering. So sometimes they do start off with extra information. So you want to add the gene, be careful on that one. You want to add the gene for ADA. The gene that you want, the gene that's going to code for ADA, needs to be cut and inserted into a plasmid. How are you going to cut it? Are you going to talk about all those different bits of information? You need to ensure that you would mention about restriction endonucleases to cut. I don't know, MKB, I haven't seen the paper. I haven't seen it, so unfortunately I've not seen it yet to be able to answer that question. So in this case, what we've got, add DNA to a new DNA to give a new characteristic protein. So we're going to add the DNA. We've got restriction enzymes, endonucleases to get the gene. Or, as someone's rightly said, we're going to use mRNA and work backwards with our reverse transcriptase. Depends on what they give us. Combine the gene with inserting it into the vector, the plasmid. Then we need to take that recombinated vector, put it into the bacterium, and then we want to clone it. So that would be one thing in which we want to do. Now, I want to bring in a slide here that I mentioned. Excuse me. Because they haven't done much on communicable diseases. I do want to bring in somewhere where I think they may potentially go. When he talks about pathogens, now vectors, we can use vectors, and those vectors could either be a plasmid, or if we were doing gene therapy, we might use a virus. So because we know that viruses take genetic material, DNA or RNA, surrounded by proteins, and they invade the cells using their biochemistry, they will therefore reproduce rapidly, and then they can burst out or bud away from the cell using a portion of the membrane. So this is important to note just about viruses, okay? What I want to know and what I want to say to you is you guys need to know about HIV. Now, they haven't touched on the immune response yet. So if they were to talk about HIV, we know that HIV targets T helper cells and therefore reduces their number. What's really interesting is these these HIV viruses contain RNA and reverse transcriptase so that they can produce their own complementary DNA. So therefore, HIV, being a retrovirus, has all the machinery it needs to insert its genes into the cell. So therefore, what it does is it makes this complementary DNA, which will then integrate into that DNA of the host cell. And obviously you need to know about how it's transmitted. We treat it with using antiretroviral drugs because it is a retroviral pathogen. So we would treat it by giving it enzyme inhibitors. Now, that's not the way I think it's going to go. I want to look at this question now with regards to actually treating the disease through using gene therapy. Oh, excuse me. And then I'm going to come back in a second to look at viral vectors. So a plasmid could be a vector, but so could a virus if we were looking at actually treating a disease, in this case, using gene therapy to try to cure ADA-SCID. So this was approved in Europe in 2016, and you're being told the stages for gene therapy. So we're going to use blood, hemopoietic stem cells. Someone said to me earlier, how could they bring back in stem cells? So we're going to use blood, hemopoietic stem cells. We're going to take them from the bone marrow of the person who has this disorder. We're going to put a functional gene and its promoter inserted into this blood cell. And then we're going to use a single infusion. And that single infusion is going to then actually be given to the patient. So explain why a single infusion of gene-corrected stem cells is enough for you to cure the disease. Now, you should know hematopoietic stem cells are multipotent. You should know that they are located in the bone marrow. So when we inject these hematopoietic stem cells into the blood, they will therefore, thank you, go through mitotic division. If they go through mitotic division, they're going to produce genetically identical daughter cells. What spec is this for? OCR A. So multipotent means that they have the capacity to go through mitotic division and to produce specific cell types within a tissue. So hematopoietic stem cells will become, whether they become phagocytes or they become B lymphocytes or T lymphocytes, macrophages, monocytes, that's where they are multipotent. They can become any type of blood cell, but they can't become a skin cell, for example. So this is me linking gene therapy, which is part of your, I want to say cloning and biotechnology, alongside your module 2 stem cells. But also linking to manipulating genomes. So the viruses which are that we're looking at are actually retroviruses. There isn't a pattern. MK, there is no pattern, which is why you need to look at the fact that is the topics that haven't come up in the first 2 papers, and also look at where there is links between there, but it's basically just go over the topics that you struggle with the most in case they come up. But I would suggest doing the ones that you haven't been assessed on already. Yeah, so bacteriophage is a specific type of virus that affects bacterial cells, basically kills bacterial cells. Yes. Okay, so stem cells proliferate, therefore the gene is inherited, passed on. That's the way. CIE, IGCC, IA level, NXLB, IB, or they are from AQA. It just depends where I've taken them from. But I've picked specific ones that will only ever be related to your topic area that you will get. MK, if you look at my videos that I've done, I actually have got a thing that shows you my predictions on my page. The other thing to look at this, excuse me, what time went? Oh, after 9, here we go. We've got a marker gene can be used when a gene of interest is introduced into a plant. Don't forget, genetic engineering can happen in plants too. If we're going to genetically engineer plants, what are we going to do? How are we going to do this? Describe and explain how the use of a marker gene coding for a fluorescent product can show that the introduced gene of interest is being expressed. Now, again, please, please, please note, sometimes the questions aren't about genetic engineering in the fact the last one was the processes. This question, describe and explain how the use of a marker gene coding for a fluorescent product can show the introduced gene. This is actually asking you about transcription translation. So this one is asking you about, do you know that the marker gene added alongside that gene of interest? Do you know they use the same promoter, therefore they will be transcribed together? Do you know that they're going to therefore have both those proteins produced at the same time? And then we're going to use the UV light to actually detect them. So if the fluorescence has been shown, transcription has occurred for the fluorescent protein, it therefore would be the same for that gene of interest. Yes, bullet point away. No, you just need to know that they have the capacity to do that. Another thing about advantages of genetically engineering, herbicide resistance, for example, because they may go down the route of herbicide resistance. If they do, higher increase of yield, less competition, lots of different ways you can get those marking points. But I wanted to finish on, I think, what am I on? Slide 66. Yeah, I'm almost there at the end of the ones for today. I've got through what I wanted to. Human immunodeficiency virus is an example of a retrovirus. A modified retrovirus is used to insert the new gene into the DNA of the blood stem cells. So linking back to that original one, state 2 ethical considerations of using a retrovirus for gene therapy. So this is where we're going to use a vector, but then it's linking to the fact that, do you understand that these retroviruses will actually go to any of the cells? Whereas if we're trying to actually create a specific type, we want to be sure what we're doing. So we want the retroviral new gene to not go to the wrong place. We want to ensure that we're not going to cause cancer or infection or immune responses or any valid point. Yes, you would want them to have the same promoter. I have linked to the excretion and liver topic, more to the excretion I talked about and liver I talked about in last lesson. I feel like they're coming up more with nitrogen cycle. I'll see what I can do. I don't have as much time tomorrow because I've got more clients tomorrow. So I don't have as much time to do planning for you, but I'll see what I can do. Yes, I am going to go live tomorrow again. I want to just finish on this last question, guys, because I haven't done anything. I've done stuff on translocation before, okay? So please do check out my videos on translocation and my lives where I've covered translocation. But yeah, liver for deamination and nitrogen cycle. Deamination produces the urea. Urea therefore results in the ammonification to ammonium ions. So check that one out. No, I wouldn't, and I'd say that that's an answer to that question. Photosynthesis can come up linked to carbon and nitrogen cycle, but I want to just finish on this question here. So the diagram shows a model to demonstrate the mass flow hypothesis and translocation. I think this is an amazing question. This was actually a multiple choice question, but I'm going to change it into just as showing you the bits of information, what you can gauge from a picture. So the minute you've been given a picture, you should therefore be able to tell them what's happening. So you've been told that you've got water. So because I've got water in this and I can see the dark side where the water is, I know that this is representing my xylem. So X has to represent my xylem. I've there also been told that I've got a concentrated sucrose solution in Y leading to Z via W. So therefore, W must be my phloem because that's where my sucrose solution is going to be transported. I know I've got a concentrated sucrose solution, so therefore I would expect by osmosis that my water will move into Y. This will therefore create a high hydrostatic pressure. That hydrostatic pressure is going to force the fluid up through and down to where there is a dilute sucrose solution. So therefore, a low hydrostatic pressure. Therefore, I can say that Y is my source and Z is my sink. I think this question was really lovely in a picture to show you translocation. And so I wanted to just finish on that one to show you where it could go with the type of question. You're not going to get a multiple choice question, but if you were, this is what it showed you in the question. So they basically had to figure out that W was the phloem, X was the xylem, so that eliminated C and D. And then you had to know whether it was the root or whether it was the leaves. So obviously X was the leaves, Y was the leaves, and therefore from Y to Z. And I don't know, I shouldn't laugh at that, but that is actually really funny. You're not going to cancel out Amna's marking, making the grey boundary higher. Don't worry. Cows will urinate into the grass. Absolutely. Nitrogen products end up in the soil and the roots. Rest goes on. Yeah, absolutely, Grace. That's exactly it. Are they available to use somewhere? No, unfortunately, they're not available for you to use right now. I haven't finished doing my workbooks, but if you're struggling with excretion, I do have a workbook on excretion. I do have a workbook. I've got a study guide and workbook on excretion and respiration. I've got it on biodiversity. I've got it on, I can't remember what else, but you can check my website out, UK BioTutor, for that one. Produce can undergo ammonification to become ammonium ions, then undergo nitrification, good, by nitrous monas, and nitrobacter to nitrate ions, good. Nitrate ions converted to N2 by denitrifying. And don't forget about the nitrate ions being uptaken by the plant. But I have done a video on that. That was in my live last, I want to say my last live. UK BioTutor, hold on. I never plug my website enough. But if you go to my website, you will find, so ukbiotutor.com is my website, and you will find my booklets there. It's already uploaded. It's absolutely, it automatically uploads. So it's here, as is all my other lives on here as well. So I have been going live since paper 2 finished, looking at paper 3, but you can actually go all the way back to my paper 1 and paper 2 lives as well. Everything is here. Nothing gets deleted. Yeah, it has been a long day for me. But then as of, yeah, go to my live section. They should all be there. I haven't deleted any, so none of them have been deleted. They should all be there. So yeah, go to all my lives that I've done. I basically have done a live for paper 1. I did lives for paper 2, and I'm now doing lives for paper 3. But basically, it can be on any topic area. Oh, you have asked me about this. Hold on 1 second. Let me go to. I'm like, I want to show you. It's literally in my little workbook. This is my prediction. Yeah, patterns of inheritance, Hardy Weinberg, and it depends if they went down speciation with the patterns of inheritance. I'm not entirely sure. I do feel that they, in my gut, I feel like you might get a patterns of inheritance in a genetic diagram and then linked to it being like, how did they inherit that autosomal disease, recessive disease trait, for example. So what have I predicted? Manipulating genomes, genetic modification, biotechnology fermenters, translocation, ecosystems, nitrogen cycle, neuronal communication, muscles, lipids, and biological membranes. They are my predictions / educated guesses that they haven't done yet on paper 1, paper 2. So yeah. Oh, and obviously module 2 anywhere. So nucleic acids and nucleotides. But I haven't seen paper 2, so I couldn't really say. How is what related, Carlo, to cloning organisms? They brought up animal cloning, which was on my list on my previous ones. I did say animal cloning was going to come up on my previous list. This is my previous list. Can you list them again? Yeah, give me a second. I had to buy myself a new booklet because my workbook's almost finished now. Manipulating genomes, genetic modification. Hold on. Let me see if I can get it on here. Give me 1 second. Guys, there's very few in you here. I'm going to say bye to you and now. Right. Don't give me 1 second. That's not what I want to do. This is what I want to do. Oh, excuse me. That is my predictions / educated guesses that I feel are going to come up. You are very welcome, guys. Thank you for staying with me. It's been a long 2 hours, so thank you very much. Nucleic acid nucleotides actually is not on this list, but yeah, I do feel like, but that kind of comes in under, so nucleic acids is kind of a given one for that one and for that one, because if you don't have the nucleic acids, you can't do any of the stuff. You are very welcome, guys. Good luck. Keep going with your revision. Keep going. You've got this. You are very welcome. Thank you for joining me. Thank you for sharing. Thank you for liking and letting people know. Hormones, I've already covered hormones, which are linked to the lipid section. Sounds ridiculous, but hormones is here. And then neuronal communication. I would have wasted these 2 hours if it wasn't. Well, I'm glad I could be of service. That's kind of why I started doing my YouTube channel, because I know I can go 4 hours and be like, how is that time just disappeared and what have I watched? Whereas actually this might help some of you. There you go. That's a really good actual suggestion. It's on there. You can go and watch it. It will stay on there. Go into my playlist, which is my live playlist. You'll be able to see it. You are very welcome, guys. All the best, and I will see you all tomorrow. Bye.